Hi everyone, my name is Fizza. I'm a first-year grad student in informatics at Illinois, working with Professor Caetano Anolis. And this is the project that we have been working on at Blue Waters, that is mining the evolutionary dynamics of protein loop structures and its role in biological functions. Now, this sort of overlaps with my dissertation project. So this is a work in progress at the moment, but I'll be presenting what we have done so far. So you might wonder that how we are linking two disparate fields such as biophysics and evolution. To answer that, I would like to quote something, a quote from my literature reviews for this project. It was that evolution lies at the heart of biology, while reductionism draws biology into the realm of physics. So it is inevitable that we keep on ignoring the physics aspect from the evolution. So that's why this is the basic premise behind this project. So the objectives of our project are that the hypothesis is that flexible, unstructured regions of proteins introduce this heterogeneity that is imperative to function, as has been proven in literature previously. But what sort of heterogeneity is responsible? We need to identify that, and how evolution has been acting on it. So in order to clarify that, I'd like to state an example here of aquaporins. Now, aquaporins are proteins that transport water into the cells. So there are two basic types of aquaporins. One is the conservative type that just transport water, while the second type is that of aquaglyceroporins that transport water and glycerol. So this is the basic structure of the aquaporins, that they have these 6 transmembrane helices and these loops. So that now you will observe that in loop B and loop E there are these NPA motifs. These are the signature motifs of aquaporins. They're present in all of them and they're conserved. So there was this study in which what they did was that they swapped the loop E of a conservative aquaporin with that of an aquaglyceroporin, thinking that since the selectivity filters of the filters that judge which molecules should go, that is either water molecules or glycerol molecules, since they are present in the center here, the swapping loops should make no difference. However, when they changed the loops, it affected tetramer stability, so that consequently affected function. So this shows how loops are acting when it comes to function of proteins. So in this case, what we plan to do is that we are exploring dynamics of loops to determine what role protein function is playing, to what extent these loops are acting here. So in that aspect, we would like to identify protein functions that could be exclusive to a certain motions that could be exclusive to certain functions in the protein. So, and with that extension, we would be looking at biophysical properties. Right now, we're focusing on flexibility and fluctuations, and interpret them in the context of evolution. So I would like to give a quick review about the protein structure, since I assume that the audience comes from diverse backgrounds. So protein structure has 4 basic levels. Primary is the linear sequence of amino acids, and secondary structure comprises of how this linear sequence has these hydrogen bonding patterns that would ultimately form these helices or beta sheets or these loop regions that we are interested in. So the tertiary structure is then how these secondary structures form complex globular structures using primarily non-covalent bonding. And then the final level is that of the quaternary structure, in which how these tertiary structures combine to these subunits combine to form a complete globule of protein. So in that connection, I'd like to define domains. Domains are peptide chains that are folded to form stable conformations of a protein. So they are a basic stable conformation in a protein. There could be one, there could be many in a protein. So, and then as far as classification of proteins is concerned, proteins are broadly classified into two types. That is, there's the structural classification, and then there's the functional classification. There are many structural classifications present, but I'll be defining structural classification of proteins over here since we use that in our project. So what SCOP does is that it groups domains into fold families. Now, what fold families are is that these domains have a recent common ancestor. That is, they have high sequence identity, around more than 30%. And then there's the fold superfamilies, in which there are four families grouped together that have a distant common ancestor. Then there are folds that have fold superfamilies with common structural topology. Now, folds may or may not be related to the evolution of its members. So the second classification, broader aspect of the classification of proteins is that of how proteins are classified in terms of molecular function. Again, there are many classification schemes, such as that by Enzyme Commission, KEGG, but I would like to go into, I'd like to briefly explain gene ontology over here. Now, gene ontology describes functions at the three basic levels. First is the cellular component of the protein, as in where is it localized inside the cell, or it could be present outside the cell. And second is the molecular function, as to what sort of elemental activity that protein is performing, such as binding, catalysis, transport. Then it also classifies at the level of biological processes that these are operations that are fundamental for living units. Now, these processes have a defined start and a defined end. So now, there are many methods for classification of proteins. There are many methods of how protein evolution can be tackled. I'm going to explain how our lab goes about protein evolution. Now, this is a methodology that has been developed in our lab. The basic assumption of that is that the most abundant domains in nature are the most ancient. So we base our phylogenomic construction on this assumption. And what exactly is a phylogenetic tree? It is a collection of taxonomic groups, that is taxa, that have particular characters with distinct features or character states, as we would call it. So how do we construct these trees? The first step in this process is the assignment of four families to proteomes. Now, this for four families, we use the tree that has been created using 989 proteomes. So we have used these 989 proteomes and done full family assignments using the superfamily database, using HMM profiles in that. And then secondly, we have done calculations of genomic abundance. That is how many domains are present in, the domains are present in how many proteomes. And the third step in this process is that the character states are defined. That is, N is the most ancient and 0 is the most recent. Now, how is this coding done? It is from 0 to 9 and then from A to N. That is, it is a alphanumeric scale, because the software that we're using, PULP, accepts these values. So then we construct trees using the maximum parsimony method using PULP. Now, people often ask me about the star. There's no footnote to it. That's the name of the software, PULP star. So After constructing the tree, we calculate ages of these domains. How do we calculate ages of these domains? Ages can be regarded as node distances here. That is, they are calculated from a scale of 0 to 1. Now, the numerator of this age is the number of nodes it takes from the most common ancestor to, say, we're interested in C. You would count those internal nodes and that node as well, and then we divide it by the total number of nodes. That would give us the age of that domain. So after that is done, that is how we determine evolutionary ages. So for this particular project that we conducted on blue waters, the approach was that we used a data set from our previous publication in which we studied the coevolution, we investigated the coevolution of amino acyl tRNA synthetases with that of tRNA. So it is an extensive discussion in itself. So we'll just, for reference purposes, I'll just say that we took a data set from this paper that had identified main domains associated with AARS fold families. So along the timeline, along the evolutionary timeline. So we took those domains and then we used the architecture database loop classifications. Now this database has houses about all of the protein loops with corresponding PDB entries. So after that, we annotated the ND values taken from the previous step to those of our data set to the loops that we had identified from the architecture database. So after overlap of these three data sets, we came with 87 classifications present in the architecture database. So we put filters on those since there were many data points and we wouldn't have been able to conduct simulations on all of them. So we used the density search method. The architecture database has two classification methods. One is the density search, one is the Markov clustering. Now in density search, the loop has the loop regions have the same length. So they could be regarded as representative. We could use a representative loop to that could be used for the entire classification, compared to the Markov clustering that has similar length. There has similar loops, but the length could be varying. So as I mentioned, that there are many loops classified in this database. So what we did was that we chose the loops with the lowest p-value, and then the loop length criterion was that the loop length should be greater than 2 amino acids, and then the secondary structure length on each side should be greater than or equal to 8 amino acids. So the overall length should be nearly 40 amino acids, not more than that, because that would be computationally expensive for us. So after that, we performed MD simulations with the NPT ensemble using an AMD 2.9 and Charm 36 as the force field. So we applied harmonic restraints on the secondary structures, and we did not apply any on the loop region to study the loop motions in the setup. And we used TIP3P water models for this. So why blue waters? It's because of the computing capability. Our lab does not have the infrastructure to support this much simulations. We did around, our target was 87 simulations, but we ended up doing 73 because we ran out of allocation. And then the other thing that I would say is that they provide ample storage of temporary files. We used to have access of this campus cluster that provided very low disk space. So it was so low that the jobs would be killed in between, because it imposed a soft limit that would not let the software write temporary files. So some of our processes wouldn't even get through. So the storage being provided to us by Blue Water is enough to get our jobs done. And the impact, other than the scientific aspect of this, is that our international collaboration has strengthened. We're working on this project with our German collaborator. We have worked with her in the past, but now we have been more involved due to the Blue Waters allocations that we have received previously and this year. So we have a much stronger collaboration with her now. The key challenge in this project is the storage of the data storage of output files. We are still figuring that out, how to go about that. So the journey so far, we conducted 73 simulations of these loops, and we identified molecular functions that were associated with these loops. These are the most abundant molecular functions. So we aim to characterize motions that are associated with these molecular functions. And as an example, I would like to quote the example of the full family domain, MyF domain, which is the tRNA binding domain, in which, as you can observe here, that it has 4 candidates that have varying loop lengths, and they have different functions. One of them doesn't even have a function. So it would give us a nice comparison, especially this one, as to how these are, how the loop candidates that have function associated with them are acting compared to this. So the global parameters that we could use right now to get a quick view of what we have done is RMST and radius of gyration to get an overview of conformational diversity and that of compactness. Now, we've calculated RMST and radius of gyration over the loop structures. We have not used the secondary structures, so just to see how they have been acting in the simulation. So these are the averages of RMST of those loop regions, of those unconstrained loop regions over the course of the 10 nanosecond simulations. So then we also calculated the radius of gyration. So far, we cannot make any global interpretation based on this since our data set is quite small. But we still have a lot to do in terms of data mining, since we just stopped doing these. We just stopped doing these simulations in April. So we still have to look at each and every of these data points to see how well this goes. So I'll just skip forward this. And so the conclusion is that the basic aim of this project is that we identify fundamental principles of molecular evolution. And this is achieved by reconstructing past events. And MD simulations can be a very handy tool in that aspect. So why would we do it? It's because this would help us in advances in synthetic biology, help us engineer better synthetic cells, and in translational medicine. It would also help us to define methods to predict future evolutionary trajectories, that is, predict evolvability of viruses as to how deadly a virus is going to be in the next season when it attacks. It is not likely that it would be deadly every time, but it helps us prepare for the worst. And then another option could be treatment of viral diseases with interfering agents. You cannot really do that on human subjects. We don't know how it might make it worse. It might make it, it might work, but this is a risky domain. So another one, so the future directions is that we are going to map motion specific to classification functions, classifications of the architecture database, and then functions from the Go database based on these molecular dynamic simulations. We're also looking at doing energy analysis maybe, and of course expand the data set. So with that, I would like to acknowledge the NCSA Blue Waters team, especially the help support, who would almost immediately answer my questions whenever I would post it, and the Illinois Research Board grant, who supported my first year of my PhD, and then our German collaborator, Dr. Frauke Grader, who's at the Heidelberg Institute for Theoretical Studies, and then the members of the Evolutionary Bioinformatics Lab for encouraging and keeping me motivated. Thank you.
All right, thank you very much. I think we have time for a quick question if there are any. Peter?
Okay. So the simulations are of about 40 amino acids with one secondary structure element.
No. Yes, exactly. They're not.
To constrain the two secondary structure elements.
Yes, we have put them harmonic constraints on them, and they're in their native positions. So it's not like they're completely fixed. They are going to move a little bit, but the basic premise is that we need to see that how the secondary structure is impinging on that loop. It could be that we are thinking of doing RMSF studies to see what if there are patterns of residues that are having these movements in a coordinated fashion. So.
Do you see a lot of movement of the sort of anchor items?
Not much, but there is. It's not like you could hold them there, so. Because simulating domains was going to be very computation expensive, so.
Give me one quick one.
Yeah, hi.
Hi.
Comment, because I know you know that the evolution of the protein has been studied from sequences by the leading evolutionary biologists by Lewis and colleagues at Yale. But also structurally, so they've been broken down. So it would be interesting when you get your root count, does it agree with all those already rather well-known evolutionary studies on the structure, the substructure, and the entire sequence?
Yes.
Look at that yet?
No, we haven't, because since this is the starting, I'm just started with this. I'll be working on it for a couple of more years. So yes, we could totally work on this. Yes, that's a valuable suggestion. Thank you.